Hello, and thank you for joining today's webinar, SubQperior, an innovative and superior syngeneic model system, presented in collaboration with Reaction Biology. Just a few reminders before we begin the presentation. This webinar is being recorded and will be made available to all registrants and attendees within 48 hours after the event. As we are all continuing to work from home due to the pandemic, we hope you will understand if we run into any technical issues or hear background noise from the speakers. We appreciate your patience and your involvement. We will start by introducing the speaker, then we will get to the content of the webinar and wrap up with a live Q&A. If you have any questions at any point during the presentation, please ask them by using the Q&A function, which you should see at the bottom of your Zoom window with the large letters Q&A. We will answer submitted questions at the end of the presentation. Next slide. So now we'll begin with introductions. My name is Megan Loy, and my main area of focus is animal welfare and in vivo services. I worked with Covance Laboratories in Madison, Wisconsin for nearly 6 years in the vivarium and client services before I moved to the Bay Area in 2017 to work with scientific software solutions. I've been with Scientist.com for over 2 years, and I enjoy working with clients and suppliers to advance the in vivo categories in animal welfare. Today, with us from Reaction Biology is Holger Weber. Holger was trained in the fields of immunobiology and angiogenesis. He is the head of the in vivo pharmacology department at Reaction Biology and has been since 2014. His scientific focus is the development of new tumor models and endpoint analysis such as flow cytometry in the field of oncology. Thank you, Holger, for joining me today.
Thank you very much, Megan, for the nice introduction. My name is Holger Weber, and I welcome you to today's webinar. I hope you felt comfortable at your computers, laptops, and smartphones and enjoy the presentation today. The topic of today's webinar is about a slight change in the injection method of tumor cells, which impacts the growth of the tumor. We have summarized this change on the basis of subcutaneous and superior in a single word. It is called SubQperior. So let's start with a short outline about the general model systems available in the field of drug development in immuno-oncology. The first 3 model systems that are depicted use the full immune system in the mouse, whereas the last model investigates test substances in mice with the human immune system and human tumor cells. The origin of these tumor cells can either be cell line derived or patient-derived. The genetically engineered mouse models express oncogenes like, for example, KRAS, or lack suppressor genes like P53, and at a certain more or less defined time point develop tumors. The advantages of these model systems are that they have a functional immune system, they have a high heterogeneity of tumor, they have a good stroma because they do growth orthotopically. You know the genetic driver in this model. The disadvantages are that you have low neoantigens, they are of high costs and challenging to set up. The modification of the GEM model is that you generate the tumor cells and implant these generated tumors subcutaneously in additional mice, like similar to a PDX model. Here, the original good stroma component is reduced, but these studies run at a lower cost and are not that challenging. Similarly to the GEMs, the induction of tumor growth is initiated in these carcinogen-induced models by carcinogens. This ends up to similar advantages and disadvantages as for the GEMs. So also here a functional immune system, high heterogeneity of tumor, but the disadvantage as well, high costs and challenging to set up. Similar to the GEMs, here also tumor cells can be transplanted to other mice in a PDX-like approach. And this you can also find on our website, where we have some mouse-derived isographs that we have generated. Finally, the model system of human xenograft models uses immune-deficient mice. Here, immune cells, by different ways, establish a human immune system. And after the establishment, a human tumor is engrafted and tumor growth is followed under therapy. The advantages here are that you are working with the immune system, a human immune system, that you can use human tumors. So it allows the evaluation of human-specific test substances. You have a high heterogeneity of tumors, especially when you use PDX models. But the disadvantages are that you have extremely high costs. It is challenging to set up. You are using a suboptimal immune system, and dependent on the route and the usage of immune cells, you might have gross versus host responses. The syngeneic tumor models are cell line derived mouse tumor cell lines. These are implanted into the mice and of the same background. So you can have a functional immune system. You have low costs and it's easy to set up. But there are disadvantages. In the subcutaneous space, the heterogeneity of tumor is very low. You have a short therapeutic window and no natural microenvironment. And most importantly, a lot of syngeneic tumor models develop at an early time point tumor ulceration. This tumor ulceration is a huge burden. There are differences between the regulations from country to country. Yet we have to euthanize here in Germany the mice at the first appearance of ulceration, independent of size, Ishar, or crater status. This is a strong restriction having an impact on the growth curve. So we were forced to search for a way to prevent or at least reduce tumor ulceration in syngeneic models. We tested different options and locations and finally end up with the SubQperior model system. But first, we have a look at the starting position. We investigated tumor growth in 12 mice for each tumor model and terminated these mice individually when an ethical termination criteria was reached. This was either tumor ulceration or tumor volume. You can easily see in this graph that, for example, the CD26 wild-type tumor model, none of the tumor reached the highest possible tumor volume because the reason for terminating was always tumor ulceration. Some models, like the B16, F10, are not as prone for tumor ulceration. But for all of those models depicted here, more than 50% of the causes for termination are tumor ulceration. So the consequences of this high ulceration rates is early termination, low tumor size, a short therapeutic window, and the need of high group sizes to compensate. When we implanted the cells in a SubQperior fashion, this model prevented tumor ulceration almost completely, as you can see on this slide. In most animal models, in this 12 mice, no tumor ulceration occurred, and only very few models show tumor ulceration. All other abortion or termination appeared when the highest tumor volume allowed was reached. So what is SubQperior? SubQperior stands for implantation of the tumor cells into the memory fat pad of mice. So it is quite a simple way of preventing tumor ulceration. The advantages are that similar to the subcutaneous, it is very easy to implant the tumor cells. You can similarly measure a tumor volume by just calibrating. You have remaining the low costs of standard syngeneic models. And finally, as I've shown, you don't have tumor ulceration. In addition to the advantages of eliminating tumor ulceration, there are further advantages to use SubQperior as the model of choice. You can see here that the growth is more homogeneous in the SubQperior compared to the subcutaneous. This results also in significant results with less animals per group. In addition, you see an improved growth. For example, here the HEPA-1/6 doesn't grow in the subcutaneous space, but you do see a growth, even so it is heterogeneous, but there is growth seen in the SubQperior environment. So tumor cells without subcutaneous growth do grow SubQperior. And finally, we checked the immune checkpoint inhibitors in models that have shown efficacy in the subcutaneous when we tested subcutaneously. And you can see in the SubQperior, this inhibition is also seen, yet with a lower response compared to the subcutaneous. The LL2 similarly does not react to anti-PD-1 or CTLA-4 treatment. So similarly to subcutaneous, to the subcutaneous model, immune checkpoint inhibitor inhibition can be compared. The SubQperior studies feature high reproducibility. Here it is depicted the response of an anti-PD-1 treatment compared to the vehicle control, and the response is in a narrow window between 60 to 80% TC. Here, the response to the immune checkpoint inhibitor can be followed among the different models that are established here in the SubQperior at Reaction Biology. And you see that the inhibition is not as strong as seen for the subcutaneous setting. That's what I've also mentioned before. This might be considered as a disadvantage, but I would say it is an advantage. Provocative, one could say, only drug candidates which make this model system respond will make it to the clinic. Another aspect is the combination testing with known checkpoint inhibitors. In this model system, there is enough room to improve the single agent response. All our syngeneic model systems are analyzed by flow cytometry to understand the immune cell populations present. We have set up a 17-marker flow panel to distinguish the most relevant immune populations in a single staining. Why is this needed? In case of a nice test substance response, the size of the tumor is limited and sometimes only one staining possible. Furthermore, with a SubQperior gating strategy, the all-in-one staining gives more accurate results. With this 17 marker panel, you can distinguish all major immune cell populations at once that are present in the tumor. So this comprehensive immune cell profiling is shown here for the clonAIM3 syngeneic model. This syngeneic model is inhibited by anti-PD-1 treatment at around 50%. You can see here that a large proportion belongs to the lymphocyte compartment and many T cells are present. The activation of the T cell compartment by anti-PD-1 treatment could be the result of the inhibitory effect that is seen. On the other hand, the myeloid compartment with macrophages and myeloid-derived suppressor cells is reduced. The comprehensive immune cell profiling shows that already with the existing syngeneic model systems, we do have a variety of immune cell populations appearing and reflecting different immune starting positions. As mentioned in the slide before, clonAIM3 has a large T cell compartment. In the Ranker model, only few lymphocytes are present and granulocytes dominate. AB12 has also many lymphocytes present, but different to the clonAIM3, it is not inhibited by anti-PD-1 treatment. And you can go on and on with the other models. Besides the relative appearance of immune populations, one should also have a look at the fraction of leukocytes per se in different models. Here, large variations are obvious, starting with, for example, the B16F10, where there is only little living leukocyte population present, up to almost 50% of leukocytes present in the fraction of single tumor cells isolated from AB12 SubQperior tumors. In a case study, we compared the immune cell populations in the subcutaneous, SubQperior, and orthotopic setting of the CD26 tumor cell line. The investigation was performed early in tumor development at day 4, 7, 10, and 13. In addition to baseline, CTLA-4 treatment was applied and the response investigated. You can download the poster from the website under the immune oncology models in the flow cytometry panel part. The immune analysis revealed that different to the subcutaneous setting, a large proportion of leukocytes is already present at the beginning of the tumor growth in the SubQperior. Here it is written intramammary fat pad, EMA. This is very similar to the finding in the orthotopic approach. In the beginning of subcutaneous tumor growth, only a low number of immune cells is present. At day 4, the tumor is very small, and for the SubQperior, it cannot be excluded that mammary tissue is also present. Yet, it shows that in the surrounding, a large number of T cells is present. The proportions of the immune populations vary a lot at the different time points tested. For example, myeloid cells make around 50% at day 7, which are then proportionally reduced by the increase of macrophages, T cells, and NK cells. These changes appear similar in all tested settings. One interesting point is that in the subcutaneous growing tumors, no B cells were detected at all, whereas in the SubQperior and orthotopic setting, B cells are present, not in all samples, but still frequently. In summary, I hope I could convince you that the SubQperior syngeneic models have advantages. Higher final tumor volumes allow the differentiation. The advantages are that you can caliper the SubQperior cells similarly to the subcutaneous, that you don't have tumor ulceration, so it is a reducement. You have a higher homogeneity in tumor growth, so as another way to reduce the group sizes. You end up with higher final tumor volumes and a longer therapeutic window, and this still at low costs. So in summary, I hope I could convince you that the SubQperior syngeneic models have advantages. Now I switch from the SubQperior model system to a more general description of steps in an immune oncology program. Here I depicted 6 steps that are of importance, and so one should start with the PK/PD, identify a responsive tumor model, depict the mode of action, confirm and improve this mode of action, test combination therapies, and verify this in a selected humanized mouse model. So a good start, which is often neglected, is to find out basic properties of the test substance in the mouse system. With your in vitro results, you get a first idea which concentrations are needed to achieve specific effects of your test compound. A good guess is to reach 10-fold of these concentrations in the bloodstream of the mice. So if you can reach this concentration in a PK study, you can move on to see changes in the mouse system that allow drawing conclusions from the activity of the test substance on its target. If you cannot reach high bloodstream concentrations, testing other formulations or administration routes might be an option. The PK/PD study should be followed by a tolerability study. Here, the question is if the treatment regimen is tolerated by the mouse, or more specifically by these mouse strains that are used later on. If side effects occur, test lower concentrations, change administration routes. In case no side effects are recognized, a treatment regimen has been identified to start working with for your efficacy study. For efficacy testing, a panel of models do exist, like I've shown before. Which model now shall we start working with? In case enough information is available about target expression, immune status, or the effect of similar immune checkpoint inhibitor inhibition, a rational selection of a suitable model can be done. In case of efficacy, a syngeneic workhorse model has been identified. If the availability of information is poor, a nice and cost-effective way is to perform a model screen. Select several different syngeneic model systems and check for tumor growth inhibition. You might at this point already include flow cytometry analysis. At Reaction Biology, we do offer such a screening. Reaction Biology provides the vehicle control group and an anti-PD-1 group and is collecting test substances from different clients, which will then run in parallel. At Reaction Biology, you can select 6 models in case you might have already got data for a specific model out of the 8 models depicted. The client only pays for his group, but receives also the data for the vehicle and the anti-PD-1 group. This lowers the cost, and in the more or less parallel screening, the results are generated fast. After the identification of the syngeneic workhorse model, an intermediate step could be done. Testing the efficacy in the model in immune deficient mice. If efficacy is seen, one might reconsider the workhorse model as the direct anti-tumor effect of the test substance and the effect on the immune system cannot easily be separated. Having no inhibition in immune deficient mice gives a strong hint that the immune system is responsible for treatment inhibition. To find out the mode of action is not an easy task. Of help can be PD studies identifying the immune cells relevant for the activity, studies which deplete relevant immune populations by antibody treatment, which in case of relevance would result in less treatment activity, or finally immune cell populations that might be isolated and ex vivo studies performed to confirm the mode of action. In this step, you should find out the immune status and can already identify biomarker and stratify them. Reaction biology can be of help to identify relevant immune populations. As I've shown before, we have established the a variety of staining panels that support the identification and are always open to establish new panels on request. The all-in-one flow panel has already been shown. Here, changes in the major immune cell populations are revealed under treatment. The other staining panels focus on different immune cell populations. Whereas the all-in-one flow panel only determines the presence of cells, the following stainings also give information about the activation of immune populations. Here, NK T cells are investigated. Several activation markers are included, like PD-1, the proliferation marker Ki67, and the interleukin-2 receptor, CD122. In addition, the memory status of CD4 and CD8 T cells can be illustrated. The next panel focuses on T cell activation. After stimulation for 4 hours with the unimycin and PMA, the cells are stained. The expression of granzyme B found in granules released to destroy target cells, interferon gamma as an activator of macrophages and inducer of higher expression of MHC2, CD107A, a marker of degranulation, and CD69, an early activation marker, are tested. And finally, dendritic cells. Here, conventional dendritic cells, CDC1 and CDC2, are differentiated, and in addition, plasmacytoid dendritic cells. PDCs are known to secrete interferons critical for the activation in viral immune responses. Having found out the mode of action by using, for example, these flow cytometry panels, it is a good idea to confirm and improve the mode of improve the efficacy of your test substance. So one could adapt the treatment regimen and see if the efficacy can be improved. If this is the case, one could identify other syngeneic models with similar characteristics and see if also their efficacy can be seen. This could, for example, also be the second choice from the screening approach. If now, after improvement of the treatment, efficacy is seen, one has confirmed the hypothesis of the mode of action. There will now most probably no test substance work on its own in the field of immuno-oncology and be sufficient. Therefore, combination testing is very important. Knowing your mode of action will help to rationally select a combination treatment. Testing it will reveal then if there are synergistic effects and help also for later testing in clinical phases. Finally, your results might be worth testing in a humanized mouse model. With the help of all collected information, design a humanized tumor model setting and perform an efficacy study. Having an effect, you won the jackpot. The way is paved for toxicity studies, and finally IND filing is possible. Seeing this picture, sketching a theoretical IO program appears already very complex. And in practice, there are many switches, obstacles, and loops. Yet, having a great target and a gorgeous test substance addressing the target, it is worth going this way and tackling the hurdles. With this, I would like to end with today's webinar. I would like to thank all the people that are listed here that help in performing the studies, performing the flow cytometry. And I would like to thank you for your listening, and let's discover together.
Thank you, Holger.
Yeah.
That concludes this portion of the presentation. Thank you for following along, and I hope this has given you some insights into how the novel SubQperior tumor models overcome common challenges of subcutaneous tumor models. We can now get into some of the questions that you have all asked. Please feel free to continue entering your questions into the Q&A section, and I will get to as many as we have time for. The first question for Holger is that you've nicely shown that the new technique prevents tumor ulceration in syngeneic models. Does that also apply to xenograft models? And if so, have you tested this?
Up to now, we have not tested that many xenograft studies when implanting into the memory fat pad, but we did have some where we observed also tumor ulceration when implanting subcutaneously. And in all of those we have tested, the tumor ulceration was prevented. And similarly to the syngeneic models, the tumor growth appeared more homogeneous and stronger than for the subcutaneous.
Thank you. And another question, you mentioned ex vivo studies. Could you give some examples of assays you've performed?
So we're here at Reaction Biology just at the beginning. We have done for customers, for example, an NK degranulation assay, or what we're doing together with a client. We do have a hollow fiber model that we use in vivo, where we fill straw-like fibers with tumor cells and implant them into the mice. We can use those fibers also for in vitro testing, and we brought in one approach together NK cells and tumor cells, and added a test substance, which then leads to a reduced tumor cell number. So these are our first approaches to also offer ex vivo analysis. And we are always open to talk to our clients to see if we can be of any help with our experience to set up ex vivo analysis.
Thank you. Another question, do the mouse models have humanized immune systems?
I didn't get.
Sorry,
the mouse models.
Yes, do the models have humanized immune systems?
The SubQperior models, these are syngeneic models. They are solely based on the mouse immune system.
Thank you. I have another question for you. Is there a correlation between quickly growing tumors and ulceration so that those slow growing tumors are less prone to develop ulcerations?
There is some correlation between tumor size and tumor ulceration, but this is not 100%. So also very small tumors can overnight become ulcerated. So it is not only dependent on the speed of the tumor growth.
Thank you. Is the implantation into the mammary fat pad really that easy, and can it be learned by like any technician?
Yeah. So in principle, the technique is as easy as the subcutaneous. Of course, you need some practice. And during the time we're now doing this, we also have found some techniques that help being 100% sure to be in the mammary fat pad. But in principle, yes, that is can be easily learned by technician or people skilled that have the skills to work with animals.
Excellent. And it looks like there's one final question. How can we access your services, and are they available on scientist.com?
How you can access the...
Oh, excellent. So it looks like we can contact you at any of those email addresses, and then you are also available on the scientist.com platform.
Yes, we are available there, and you can, of course, always contact us over the website. And
Thank you.
We will respond immediately.
Excellent. Thank you so much. And thank you all for joining today's presentation. If you have any additional questions or you'd like to get in touch with our team at scientist.com or Reaction Biology, that information is available on this slide. And you will each receive an email with further information about the topic presented, including the recording of this presentation. Thank you to Reaction Biology for their involvement in the presentation, and thanks again to you for joining us. And we hope you found this presentation useful.